mismatch, improperly base pairing with the complementary strand. When such a mismatch occurs, DNA chain elongation generally pauses. This pause allows spontaneous melting of the end of the DNA strand being synthesized, freeing the 3' end with the mismatched base to enter a 3' exonuclease site on the DNA polymerase. The exonuclease site catalyzes the removal of several nucleotides from the 3' end of the growing strand. Only the removal of the nucleotide carrying the mismatched base is shown here, though additional nucleotides would actually be removed. Following removal in the 3' to 5' direction, DNA polymerase can then resume its synthesis activity in the 5' to 3' direction. The proofreading function reduces the overall error rate to as low as about 1 in 100 million for DNA polymerase 3.
So that's pretty effective. It changes it from an error rate of 1 in 100,000 to 1 in 100 million. And that's the our DNA polymerase proofreading function. The things that can cause mutations include mutagens as well. So mutations can happen just by mistakes in replication, but also mutagens. And that can be environmental influences, such as energy, radiation, UV light, or chemicals. Any chemicals that can cause mutations are called carcinogens. They have a special name. The rate is low because DNA repair enzymes also repair and make any changes to DNA as errors happen. Okay, and that happens in the nucleus. Another unique way of disrupting genes and chromosomes is transposons, jumping genes. This is where sections move to new locations and disrupt sequences. So take a look at this. This is a bacteria, and it's got a loop of DNA as its chromosomes. So instead of 46, it just has one single loop. But that's a eukaryotic cell. That's a living thing. And over there is a section of DNA. It's called a plasmid. It's a ring, usually associated with viruses. So take a look at how this disrupts chromosomal DNA.
Transposons are segments of DNA that are capable of shifting from one location to another. A transposon enters the cell by being carried on a plasmid. A transposon can then move from the plasmid into the host cell genome. A transposon can move from one site on the host genome to another site on the host genome. When some transposons move, they replicate, leaving a copy in the original position. A transposon can also move from the host genome to a plasmid.
So an example of this, and we would have seen it in nature, is maize and corn. Corn and maize are traditionally, historically purple, but we nowadays buy our lovely yellow corn to eat. And that arose from these jumping genes interrupting the purple genes so that you get this marbling. And then through genetic modification or selective breeding, we've taken away all of that purple and now our corn is yellow. But you'll often see this as decorative corn or those specialty corns with purple in them still. And that's caused by transposons jumping in and interfering with the expression of the purple gene. So those are a couple of ways that mutations or disruptions in DNA can occur. There are two broad types of mutations. The first one is called chromosomal mutation. And this is where large segments of DNA are changed, so much that we often see the effect when we look at the karyotype of an organism. So we can see physical differences to the chromosomes because of these mass changes in DNA. So the first one is the deletion or addition mutation, chromosomal mutation. And this is where a segment of chromosome is added or deleted. So you can see there's 22 other matching pairs, and then this one pair has this segment cut out of one of the matching pairs, so the homologous pairs. So this has been shortened, and if we took a karyotype of this, we could see a structural difference to this chromosome here. A duplication one is similar in that instead of cutting a section out, a section of the chromosome has been repeated for some reason, and again, a physical difference that's observable there. In inversion, this one is a little bit less easy to spot on a karyotype, but what could happen is a section of DNA could be cut out and then inverted and reinserted in. And that could disrupt any of those genes that are in there that need to be expressed. Another one, you have 21 pairs, and then this is 22nd pair and 23rd pair if this was human, and a section of one chromosome is cleaved and then inserted into a neighboring one. And you can see that it has an effect on two chromosomes, not just one. An example of some of these chromosomal mutations are Charcot-Marie-Tooth disease, and this one is where we have some duplication of chromosomal material in the short arm of number 17. So we have some duplication of genetic material here. Okay, it leads to some neuropathy, so some nervous issues in the fingers and toes as you get older, usually appearing when you're an adolescent into young adulthood. And this is seen in about 1 in every 2,500 humans. Smith-Magenis syndrome occurs in 1 in 25,000 people, and this is deletion of genetic material in the short arm of chromosome 17. So not duplication, but deletion this time. So deleted, deleted. All the other chromosomes are fine. Usually the outcome of this is small stature, but engaging personalities and some other behavioral disorders associated with that. So overly friendly, don't know where boundaries are, tied to physical and cognitive delay as well. Williams-Bjorn syndrome, a little bit similar to the last one that we saw, appears 1 in 7,500 to 1 in 20,000 births. And this is a deletion of 26 genes in the long arm of chromosome number 7. So this is chromosome number 7, so there's a deletion of a bunch of material there. And you can see there's some characteristic physical attributes, elfin appearance, low set ears, short stature, upturned nose. These disorders usually bring on cheerful demeanor again, very friendly, don't know where boundaries are, physical and cognitive delay as well. Another type of chromosomal mutation is nondisjunction. And this happens when cells are splitting and the homologous pairs are supposed to divide to the two new cells. And instead of everything being split evenly, what happens is one of the cells gets too much genetic material while the other one doesn't get enough. So here's an example of a cell. It's supposed to have 46, but we only see 2 chromosomes there. So it replicates its DNA in preparation for cell division. And when the cell division happens, instead of the one black arm going here and one black arm going here, and same for the white arms, both black arms end up over here. So this cell would have 47 chromosomes, while this one has 45. Now, if this happened in a skin cell or a hair cell, maybe that cell would just die. And that's fine. You have thousands and thousands of cells dying at this very moment, and they're being replaced by cell division. In fact, if you're growing, like most teenagers, you would be producing more cells than are dying. But this is more problematic if this was happening in a sperm or an egg, a sex cell. We know that there's 2 phases in meiosis, so this would be phase 1, and then phase 2 in meiosis would be a splitting of this and a splitting of this, so that instead of having 46 chromosomes each, it would have 23. In this case, you can see if this splits up, this one would go with a black and white, and then the next one would only have a black. This one would have 1, and then the fourth sperm or egg there would have none. So if that was the case, you would have sperm with 24 chromosomes, 23 chromosomes, 23 chromosomes, and 22 chromosomes. Now 23 is fine. The 23 chromosome egg with a 23 chromosome sperm fused to make a full complement of 46 chromosomes. But if you have a 24 chromosome egg with a 23 chromosome sperm, you have 47 chromosomes. And if you have a 22 chromosome egg with a 23 chromosome sperm, you end up with 45 chromosomes. So in those two scenarios, 47 and 45, there's a problem there. Now you can see I mentioned that it happens with the egg, and that's most often the case because as women get older, their cell division, their meiotic cell division, tends to make more mistakes. It may happen the same as in men, but the fact that men produce millions of sperm and it's a bit of a race for those sperm to reach the egg, usually a defective sperm wouldn't make it to the end. We all know of an example of this. It's called Down syndrome. Down syndrome is actually called trisomy 21. In this case, trisomy 21 is where you have a triplicate of chromosome 21. This pink one came from dad. These two came from mom's improperly divided egg. So as a result, one egg got no chromosome 21 and one egg got both chromosome 21, and then dad fertilized it with an extra 21 chromosomes. So now we call that trisomy 21. So there's 3 chromosomes of 21. And that results in Down syndrome. And we know, we've probably seen, or you probably know someone with this syndrome. And it's characterized with round moon face, flat features, missing some of those folds and things like that would give you expressions, slanty eyes, upturned nose a little bit, low set ears, widespread toes, things like that. Obviously, cognitive and physical delay as well. As you can see, in trisomy 21, the rest of the chromosome pairs are fine. It's just this one. There's other ones, trisomy 18, also known as Edwards syndrome, and trisomy 13, Patel syndrome. Down syndrome, 1 in 300, which is quite common. So McGee being 900 students, we'd expect around 3 people born with trisomy. And Edwards syndrome, 1 in 1100, and Patel syndrome, 1 in 3000. Now, we can test for this because all we need to do is figure out if the fetus developing in the uterus has a full complement of chromosomes or too many or too few. So if you're an older mom, 32 or older, we often suggest that you do this test where we take a needle, we shove it into your belly, and we suck out some amniotic fluid, and we find a skin cell from the child, and we work up a karyotype, and then we just count the chromosomes. Now, Down syndrome, trisomy 21, is supposed to be as frequent as 1 in 300. We don't see that so often in developed countries because in Canada, at least, we can have the fetus terminated by the choice of the family. In other countries, third world countries, and other countries that don't have the appropriate laws, they would have to carry them the full term. So their occurrence of trisomy may be more common. Edwards syndrome, that's one of the trisomy, I believe it's 13. Upturned nose, low set ears, it's a really small jaw. You can see the clenching of the hands and the rockering of the foot. Usually small stature gain, physical and cognitive delay. Patel syndrome, you can see the cleft palate, the low set ears, small head, absent eyebrows, funny clenching of the fists, low set ears, and small. Now, in addition to trisomy of the non-sex chromosomes, it can happen that we can have nondisjunction of the sex chromosomes. So, in this case, it's called Turner syndrome. When mom was making her eggs in her body, okay, not under her control, just natural, 2 of the X chromosomes went to one egg, and no X chromosomes, no sex chromosomes went to another egg, and that was the one fertilized by dad's sperm that had an X chromosome. So as a result, instead of having 46 chromosomes, there's 45. All the other chromosomes are paired up with their right pair, and then you have this one, which doesn't have a pair. Because it's the X chromosome, this person would present as female, but would be sterile, probably would suffer from cognitive and physical delay. And this is often as common as 2,000, 1 in 2,000, or around 1 to 5,000 births. And sterile, so no menstrual period for this person. Klinefelter's is the opposite. Instead of getting the egg with only getting the egg with no X chromosome, this is the one where the egg has both X chromosomes, and then dad, fertilizes it with Y chromosome, which makes sense, because dads can only be the one giving up Y chromosomes. And this is a bit like trisomy in that it has a triplicate or triple set of chromosomes for the sex chromosomes. This person would present as male, probably sterile, but would have finer muscle tone, smaller stature, physical and cognitive delay as well.